Section 15.1, Mendelian inheritance has its physical basis in the behavior of chromosomes. Our first topic is the chromosomal theory of inheritance. This theory places its roots in 1875. It was at this time that technology had advanced enough to improve microscopy techniques, allowing scientists to work out the process of mitosis and meiosis. Biologists began to see parallels between the behavior of chromosomes in these processes and the behavior of Mendel's proposed hereditary factors. During meiosis, scientists noted that both chromosomes and genes were present in pairs and diploid cells. We know that this is because the genes are attached to chromosomes, which form homologous pairs. These homologous chromosomes separate during meiosis, and the genes on each chromosome segregate. After fertilization, each pair of chromosomes was restored, and their genes once again matched up. It was this discovery that formed the chromosomal theory of inheritance, which states that genes occupy specific locations or loci on the chromosomes, and that these chromosomes undergo segregation and independent assortment during meiosis. This theory was discovered in 1902 by Walter Sutton, Theodore Boveri, and several other scientists, all separately. This theory can then be proven in Thomas Hunt Morgan's experiment.
Sutton and Boveri are two Greek scientists who established the Sutton-Boveri chromosome theory. Without their hypothesis to strengthen Mendel's statements and experiments, no one would have understood how people have different eye color, skin color, height difference, and other unique features.
Okay, this is one of Mendel's dihybrid crosses. We started with the P generation, also known as the parent generation. We started with the yellow round, true breeding yellow round, which is big Y, big R, crossed with a green wrinkled, which is little y, little r. And the result of the result from the first generation, we got phenotypic ratio of 1/4 big Y, big R, 1/4 little y, little r, 1/4 big Y, little r, and 1/4 little y, big R. After crossing the F1 generation with the F1 generation, you'll get the phenotypic ratio for the F2 generation, which is 9:3:3:1. You will get 9 yellow round, 3 green round, 3 yellow wrinkled, and 1 green wrinkled. This is the exact cross that Mendel did. As you can see here, it shows you clearly the F2 generation of how they got 9:3:3:1. The 9 round yellow, the 3 round green, the 3 wrinkled yellow, and the 1 green wrinkled.
I'm going to teach you guys about the law of segregation. During the production of gametes, the two copies of each hereditary factor separate from each other. This is why you get one trait from one parent and one trait from the other. This happens during anaphase 1 of meiosis. And then there's the law of independent assortment. This is when the alleles for each trait separate and then they are randomly put back in a different spot on the homologous pairs. This is when different loci separate off their homologous pairs and they go on to other ones, so the traits get separated and split up so everyone looks different. If this didn't happen, we would all look exactly like our parents.
Okay, this is the chromosomal basis of Mendel's laws. Here we're going to correlate the results of one of Mendel's dihybrid crosses with the behavior of chromosomes during meiosis. The arrangement of chromosomes at metaphase 1 of meiosis and their movement during anaphase 1 account for the segregation and independent assortment of the alleles for seed color and shape. Each cell that undergoes meiosis in an F1 plant produces 2 kinds of gametes. This is why all the different pea seeds don't look the same, or they're all different. And this is why the dominants and the recessives will all switch with each other. If we count the results for all cells, however, each F1 plant produces equal numbers of all 4 kinds of gametes because the alternative chromosome arrangements at metaphase 1 are equally alike.
The first evidence associating a specific gene with a
specific chromosome came from the work of embryologist Thomas Hunt Morgan. Morgan selected fruit flies for his experiment because they only had 4 chromosome pairs. After 2 years of mating the flies, eventually he had a breakthrough. Instead of having the usual red eyes, a male fruit fly had white eyes. Red eyes are wild types because that phenotype for a characteristic is most commonly observed in populations. And the white eyes are called mutant phenotypes because they stray away from the wild types.
Yeah.
Hi, Thomas Hunt Morgan was the person who conducted an experiment to show how some genes can be sex-linked. He conducted this experiment with fruit flies. What he did was he took white-eyed males and he crossed them with red-eyed females. Now at the time he knew that red eyes was the dominant gene. So when he crossed the two, for the F1 generation he got all red eyes for both male and female. Then when he got that, he crossed the F1 generation with other F1 generations, and he got a 3-to-1 phenotypic ratio. This was odd because what was supposed to happen was the females were supposed to be half red and half white, as well as the males. But what actually happened was the females turned out to be all red, while the males turned out to be half red and half white. So what the experiment proved was that some genes can be sex-linked, such as eye color in flies.
Go.
Okay, so this is Thomas Morgan's experiment. He started out with a white-eyed male and a red-eyed female. Now remember, the red eyes is a dominant trait. Then he crossed those, and he got all red-eyed offspring, which were 2 were male and 2 were female. Then he took the F1 generation and crossed them together to get an F2 generation, which created a phenotypic ratio of 3-to-1. He got 3 offspring with red eyes and 1 offspring with white eyes. The white-eyed offspring was the abnormality. At the time it was called the mutant gene. So in conclusion, what the experiment in the end concluded was that this gene is sex-linked, because what was supposed to happen, if you follow the cross, was that the 2 males were supposed to be like they are right now, 1 red-eyed and 1 white-eyed, but the female was also supposed to be 1 red-eyed and 1 white-eyed.
This is a video we found that will wrap up our entire section.
Alright, ladies and gentlemen, well, in this lecture what we're going to do is look at the relationship between the behavior of movement of chromosomes and the passage of traits from parent to offspring. Now, we know that the traits we see in an organism, looking at a person, the color of their skin, color of their eyes, color of their hair, the height of the individual is all due to the production of proteins that build and then operate the body. Now, we know through the central dogma that genes are the instructions for making proteins. So, thinking of the central dogma, we ultimately take this information, and use it to make the proteins that build and operate our body. Now, genes have a physical location, and that location is on the chromosomes. So, by observing the movement or tracking the movement of chromosomes during meiosis, you are able to elucidate or see how genes, in essence, get passed on from parent to offspring, following those patterns first observed by Mendel. Now, this is a nice diagram that goes into images related to meiosis. So, here we have, production of a new organism, and then voila, these individual cells are going to be undergoing meiosis. Now, Mendel's law of independent assortment is illustrated by the relative positions or arrangements of these homologous chromosomes. if you have a particular set of chromosomes from one parent, they can both be lined up on the same side of the metaphase plate, or they can be on opposite sides of the metaphase plate. How one set of homologous chromosomes pairs up has no impact on how a different pair of homologous chromosomes arrange, because again, they're different spindle fibers attaching to the different homologs. Now, the law of segregation is illustrated by the events of anaphase 1. To segregate means to separate. So here we have these homologous chromosomes, one from one biological parent, one from the other. During anaphase 1, as the microtubules shorten, as the motor proteins walk the chromosomes down the microtubules, you physically separate homologous chromosomes, so that ultimately what gets passed on in the gametes is only one copy of the original pair. So in the diploid organism, you have two copies, one from each parent, but then again, in the offspring, only one pair is passed. So again, by tracking the movement of these chromosomes, we are able to confirm Mendel's laws of independent assortment and segregation. Now, much of the early work on chromosome behavior was done in the lab of Thomas Hunt Morgan, and he did his work using fruit flies as a model. Fruit flies were chosen because of their quick generation time. They can have a new generation created within about 2 weeks. They produce a prodigious number of offspring, and they only have 4 chromosomes to track rather than the 46 that we see in humans. Now, there are a couple of important terms here that you'll want to review. The first being wild type. Now, rather than saying dominant or recessive, the lab that Morgan was in looked at whether or not a trait is typically seen in nature or, in this instance, rarely seen in nature. Now, the trait typically seen in nature is referred to as being wild type, so just think of what would you see out in the wild. Now, in the instance of these fruit flies, the wild type characteristic is red eyes, so there's a protein produced that gives the eyes these red color. Oftentimes in nature, a mutant form of a gene will lead to a non-functioning gene and ultimately no protein being produced. So in this instance, the white eyes are the result of a mutation that prevents the production of the red pigment. Oftentimes in nature, a mutant form of a gene will lead to a non-functioning gene and ultimately no protein being produced. So in this instance, the white eyes are the result of a mutation that prevents the production of the red pigment. So again, to review, wild type produces the protein. We oftentimes refer to this as being dominant. The mutant type produces no protein. We often think of this as being the recessive form. Now, the nomenclature they use, or the symbols they used the first letter of the mutant allele, so they chose W. And to indicate that for this particular W gene, as they refer to it, the wild type produces the protein, so they give a positive sign or a plus sign there to indicate the wild type gene. Now, looking at the experiment conducted by Morgan, he originally took two homozygous, a red-eyed female homozygous for the wild-type gene and crossed those females with white-eyed males, so they're mutant for that particular gene. Now, obviously, if the parent is homozygous wild-type and the other parent is homozygous mutant, all the offspring are going to be heterozygous. So heterozygous offspring in this instance had the wild-type characteristic. They were all red-eyed. But what they did was allow males and females from this F1 generation to do was breed with one another. As a result of that, the F2 generation had red and white-eyed organisms. But what was unexpected was the fact that females only had red eyes, and males, approximately half of them had red eyes and approximately half had white. So while the genes were passed on from parent to offspring, the passage of those genes was somewhat unexpected. Now, Morgan worked through this conundrum here and proposed that what must have occurred is that this particular gene for eye color is found on the X chromosome. That's significant because males and females have different number of X chromosomes. Since females have 2 X chromosomes, they'll be affected at a different rate than males in relation to the reception of this gene for red eyes. Now, at this time, I'd like to fold in the term hemizygous, hemi meaning half. Males are referred to as being hemizygous for sex chromosomes since they only have 1 X. Now, this is a nice image of what occurred in the lab experiments done with fruit flies in relation to this eye color. Now, if we look at the example here, mom is, again, homozygous for wild type, so she has 2 of the W plus alleles. And dad is mutant for that allele, so he has the W with no superscript there. And again, since it's on the X chromosome, dad only has 1 copy. So in effect, the phenotype of the father is white-eyed and the phenotype of the mother is red-eyed. Now, when they mated, mom, being homozygous, always passed on the wild-type allele to her offspring, and dad, being mutant for that gene, passed on the X chromosome with the mutant allele to his daughter, but to his son he passed the Y chromosome, so it's unaffected. So the offspring of this mating then were able to mate with one another in the F2 generation. Now, in this F1 they all have red eyes, right, because each of them has a wild-type allele that they inherited from their mother. So the father in the F2 generation, then, he can pass on his wild-type allele in the X chromosome to all of his daughters. To his sons, he'll pass the X chromosome, so that's inconsequential for this particular trait. The mother, being heterozygous, to half of her offspring, you would expect her to pass on the wild-type allele, but to half of her offspring, she would also pass on the mutant allele. Now, this doesn't really impact the phenotype of the daughters because all daughters are going to get a wild-type allele from their dad. But since the males are hemizygous, the mutant allele from the mother would be expressed in the male, even though it's only 1 copy. So that sort of fits the pattern of inheritance seen by Morgan. All the females were red-eyed, and half the males were red-eyed, and half were